Welcome friends. In this video we'll be talking about enzyme immobilization, which is a very important technique, and it's a very modernized technique to utilize enzymes in so many reactions and to restrict the loss of enzymes in the industrial purpose. So let's discuss. Now the immobilization can be divided into two different parts. It can be of immobilized enzymes, it can be immobilized cells. So in case of immobilized cells, we take cells and we entrap them, immobilize them. In case of enzymes immobilization, we simply take enzymes, we won't take the whole cell, we just take enzymes and immobilize it. Now if we look at the enzyme microorganism and cell immobilization, we can find several techniques. Now the technique, as we can see, so let me take a color first. Okay, let's go. So what we can see, there are several techniques like adsorption, which is a simple attachment of the cell surface with a matrix, and second one is adhesion. Again, it's due to any kind of utilization of bacterial flagella or cilia or pili with adhesion with any kind of matrix. Now this is the third kind of covalent binding. This type of bonding between the bacterial cell surface with the matrix is via covalent linkage. It can be via the ionic interactions or ionic bonding. Cross-linking can be there. In this case of cross-linking, we can take bacterial cells which were we cross-linked via other type of cross-linking agents. Especially the cross-linking agents used in industries are glutaraldehyde. Now encapsulation can be there. Encapsulation mode, what you can see, we produce the capsules under which or inside which we put our cells or enzymes. So whatever steps we are seeing, adsorption, adhesion, covalent bonding, cross-linking, this can be seen here in this picture for the bacterial cells or the whole cell immobilization, but these techniques can be applied to immobilization of enzymes also. So instead of the cells, we put enzymes, in those case of enzyme immobilization. Now we can also have entrapment or inclusion, as you can see here, which you produce a matrix and pores are generated in between the matrix onto which the bacterial cells or enzymes are getting to be entrapped. Now this is the enzyme immobilization processes. There are mainly three major categories of enzyme immobilization. One is carrier binding, second one is cross-linking, third one is the entrapment. Now the carrier binding is facilitated different types, like physical adsorption can be there, as we have seen the previous case. Ionic binding can be there, and also covalent binding can be there. Now remember, if we go back to the previous case, adsorption means is by means of mechanical attachments. No any kind of covalent linkage or any kind of ionic linkage is there. So it's a very small at very minute attachment, actually very weak attachment. And in case of adhesion, it can be via the covalent interaction, and also it can be via the ionic interaction. Now if you use ionic bonds, for example, say if we take the matrix of positively charged and our cell or enzyme is negatively charged, so they can be attached with each other via the ionic interaction. Okay, so this kind of bonding can be there. So in any kind of this carrier binding, remember the term carrier, that means in all the cases, adsorption or ionic binding or covalent binding, we must need a carrier for the enzyme to be attached. Now the carrier we utilize in this case must have a basic property, and the property is that the carrier must have insoluble nature in water. So it must be insoluble in water, and it can be polymerized to produce a matrix, right? So these things are very important to have for the carrier. Now the second type is the cross-linking. So let us look at the image. Here we can find the image. So this is the normal carrier binding. So this yellow thing is the carrier onto which we are having the physical adsorption of enzymes, like that. Now these red things are enzymes in this case. Okay. Now we also have this cross-linking. In case of cross-linking, we take some cross-linking agents. As you can see here, it is depicted by this yellow colored rods, and these red things are enzymes. So what we are doing here, we take enzymes, and the enzymes are linked with each other. They are linked via this cross-linking agents. Usually the cross-linking agents that are used are glutaraldehyde solution. Okay, now let's say the entrapment. Now in entrapment, what we produce lattice utilizing several matrix proteins. So several kind of agents, it can be cross-linking agents and other type of things like polystyrenes, or say it can be of starch, cellulose, and all these materials which will produce a polymerized network, like that. And the network will have gap between them. Onto this gap the enzymes can be entrapped, enzymes can be added and trapped. Okay, and another mode of entrapment can be seen by this microcapsule formation. Now in this microcapsule formation, what we produce a cellular coat under which or inside which we put or add our enzymes. So this whole shell is an cellulose coat or cellulose vehicle, okay, inside which enzymes are delivered. So let's begin with the physical absorption mode, which is the type of carrier binding type. So we will see physical, ionic, and covalent, three of them. So this is the first one, physical absorption mode. Now this mode, again, the binding is mainly by the hydrogen bonds, multiple salt linkages, or van der Waals interactions. Nothing too very special in this case. Now the advantages is that this is very simple and cheap, because it's simple attachment via the hydrogen bond or multiple salt linkages, and we all know that hydrogen bonds are weaker force, van der Waals is also weaker force, so it is a weak bond. It causes little or no conformational change of the enzymes due to the mechanical surface to surface attachment. Because suppose this is our enzyme, let me change the color, sorry. So let's see, this is the matrix. So it's simply surface to surface interaction, so no change in this interaction is happening. So it's a very weaker interaction between the enzyme and the surface of the matrix or carrier. Now the disadvantage is the absorbed enzymes may leak from the carrier, as the bond between them is very weak, so they can leak. So the enzymes can leak due to any kind of mechanical forces. Any kind of small mechanical forces can take this enzyme out of this attachment, right? It can also possible. So it will we can have that what is called the enzyme loss. Okay, now the use of this adsorption technique is in case of the vinegar production. We can use it, the adsorption of this acetobacter species, which is a whole cell, onto the wood chip. So we take the whole cell immobilization via the adsorption in case of the acetobacter species is attached to the wood chips in case of the vinegar production. Now also in case of manufacturation of the beer, what we are having, the Saccharomyces cerevisiae, it is immobilized by the adsorption and attachment to the polystyrene. Remember, I have already discussed the polystyrene is provide the matrix, or it can provide the matrix for the attachment of cells or enzymes. So here it can be used with the DAE cellulose, which will provide the matrix. Now this DAE cellulose can provide the matrix because it is a polymer. Now the second time is the ionic binding mode. Now in ionic binding mode of the enzyme, protein it actually it will attach to the water insoluble carrier. So remember, I have told you that the one property is very important for the carriers to be, that is it must be water insoluble most of the cases. So it should be water insoluble, and what it will have particular charge. For example, say here this M is a matrix of the carrier. Now the carrier is containing the positive charge, so it will bind with the negatively charged enzyme. So if you see, these are the enzymes and they are negatively charged, they are having the negatively charged surface, so they can go and attach to it because positive attracts negative, so it will attach via the ionic interaction. Okay, now usually the matrix that are prepared here, they are prepared for from inorganic materials. They can be prepared from organic materials. We will be discussing about these carriers or matrices later, but now let us see the basic way. It is made up with mostly polysaccharide residues or synthetic polymers. Now the polysaccharide residues we utilize to make these things can be of natural type or synthetic type. Natural polysaccharides are cellulose, starch, and synthetic polysaccharides are those polystyrene residues like that. Okay, now let us look at the third kind of interaction. This is the covalent binding mode. In covalent binding mode, simply the enzyme will be attached to the matrix or a carrier, which is again the of the same type or the similar kind like this polysaccharides and synthetic polymers. So the only difference between the ionic bond and covalent binding mode is, in case of covalent binding modes, the interaction between the enzyme and the matrix, or the enzyme and the carrier, is via the covalent bond. Now the covalent bond can be supplied utilizing amino group, carboxyl group, sulfhydryl group, indole group, thiol group, hydroxyl group, and so on. So the groups that are present there will provide the binding interaction or the binding residue to be attached with the enzyme. Now what are the advantages is that due to this covalent interaction, as we know covalent interaction is very strong interaction. So due to this covalent interaction, enzyme loss is very less, so no leakage of the enzyme happens. Now remember, if we go back to the previous slide, we've discussed about the disadvantage of the physical absorption. We've seen that absorbed enzyme may leak due to the very insufficient strong linkage, so due to the weak linkage between the enzyme and the matrix. But here we can see there is no linkage due to the very strong interaction between the enzyme and substrate, enzyme and the uh, matrix, which is achieved by the covalent binding. And the disadvantage is also there due to the kind of covalent linkage. We know this covalent linkage is very strong, and it is sometimes irreversible, so the covalent binding may alter the conformational structure. So this thing can happen, so it can alter the conformational structure of the enzymes. Now if it can alter the structural conformation of the enzymes in the active center of the enzyme, so due to the change in the active side of the enzymes, it can have a loss of activity for the enzyme. So this thing can happen if it changes the conformational structure of the enzymes at the active side of the enzyme. It can lead to the loss of enzyme activity, because the enzyme is acting with substrate by its active side, right? Now let us talk about the second different way of enzyme entrapment or immobilization, which is called the cross-linking. Now in cross-linking, what we do, we take enzymes and also cross-linking agents, and also carrier can be taken as a support of matrix. Now here in this case, the intermolecular cross-linking can be seen. So we take different molecules like this enzyme molecules, so they will be interacted. So this is the interaction between intermolecules, so interaction between molecules. Okay, so the interaction can be with other protein molecules or with other type of functional group which can be have a supporting matrix. Now again, in case of cross linking also, we need a supporting matrix, which is very important. Now the type of matrix here we'll be dealing with must not be soluble in water, right? So again, insoluble thing should be there, so matrix should be there. And also, so in this case of cross linking, what we have a strong matrix. And suppose we are having this red color thing, these are the enzymes, and we need to attach them together. So for the proper attachment, what we have, they are cross linking agents. So this is a type of interaction we are talking about now. They are cross linking agents, like that. Okay, so we need three different players for the cross linking. One is our enzyme, so this is our enzyme. One is our uh, matrix, so this is our matrix. And third thing is, we also require some cross-linking agents. This is the cross-linking agents, and usually we use glutaraldehyde as cross-linking agent. Now we can see in this picture that this is the enzyme and this is the glutaraldehyde, which is the cross-linking agent. It will hold on to this enzyme and take it and attach it to the matrix, okay, which is insoluble, and they produce this. Okay, now let us talk about the enzyme entrapment method. Now in this case, the localization of an enzyme within the lattice of a polymer matrix is achieved. So again, the matrix will be required. Now what is going on in this case, what we produce a polymer. Now when you produce polymer with some kind of polymerizing agents like polystyrene or polyvinyl alcohol, whatever, if you take these things, so when they are arranged to form the polymer, what they produce different lattice structure, like this one here, this kind of lattice structure, which will have the linked region as well as the gap regions. Now they produce these gap regions or pore regions onto which the enzymes can go and will be entrapped. The enzyme entrapment onto these pores can be of side to side, surface to surface interactions. Okay, so the enzymes can be entrapped there. Now what we'll do then, if we add substrate, the enzyme can act on substrate and convert it into product. Okay, so this is the thing. Now, the another mode of entrapment method is via the microcapsule mode. In case of microcapsule, it is a little different, but it is also a type of entrapment. So what we can see here, we take enzymes and put into the vesicle. We make vesicles utilizing different polymerizing agents. Again, the polymerizing agent that are utilized during the production of these droplets for entrapped enzyme droplets are, say here, natural polymers like starch, cellulose. Mainly these natural polymers are utilized to deliver these enzymes as a packet of enzyme, right? So you make a packet of enzymes in this case of microcapsule production. So we have discussed about different techniques of enzyme delivery and enzyme immobilization. Now in all these cases, in fact most of the cases, we require different carriers, and also different types of polymerizing agents which can play as matrix to hold on to the enzyme. Now we know their properties are important because without them the enzyme cannot be entrapped. Now the whole concept of enzyme entrapment is that we need some solid support. Now the support is played by these carriers, right? So these carriers can be natural, it can be synthetic or whatever, but we require this kind of support onto which enzymes are going to go and attach with. Okay, so these are the utilization in the standard method of enzyme immobilization. Now there are some characteristics of carriers are important, like the chemical one, so that the hydrophobicity of it, chemical and microbial stability of it, which is really important. Now the morphological features like the particle diameter, whether it is long rod shaped like that or circular like that or linear like that or whatever, we need to know all these things because we can use these carriers depending upon our purposes. So if we require the cell enzyme to have a whole round covering, then we cover it onto this circular circular matrix. If we require it to be entrapped, then we utilize a column and then we entrap the enzyme onto it. So depending upon our purpose, we will vary these things. So we need to know the morphological feature. We need to know the mechanical feature because this mechanical feature is going to tell us whether we can use this carrier as a what kind of immobilization procedure. Is it a kind of adsorption or ionic or whatever? Okay, now resistance to pressure, compressibility and elasticity is also very important. Now again we need the general information like the food or pharma grade and cost of all these things, because again in industrial perspective cost is a very important thing to be considered. Okay, now we can classify carriers in three different parts. It can be inorganic and can be organic. Now among the organic we can again divide into two different parts. It can be of natural sources, it can be of synthetic materials. We have already discussed it earlier. Now let us look at the inorganic carriers. Now the advantage of inorganic carriers are they high pressure stability, and also they may undergo so this is a very good advantage. So this is the advantage part. Second thing is that covalent binding carried out can be carried out in this case, so you can utilize it and they are very pressure stable and also heat stable in many cases. Now what we use generally, we use silicon dioxide here, SiO2 based carriers. Now from where we get this SiO2 based carriers? From the porous glass and silica, because the glass contains many silica as we all know. So the porous glass like waters, groaning and all these things, so gorning, we can take this porous glass which is a source of SiO2 and we can produce our carriers. Now we need to functionalize this carrier or activate the carrier, and to activate the carrier what we do, introduce amino groups onto the carrier. Otherwise then otherwise the enzyme may not be attached to it. So we need to provide amino acids to make it functionalized, then we provide the enzyme which are going to attach with this particular inorganic carrier. Okay, now we can also use mineral materials like clays, like selenite, bentonite and all these things. Okay, now cross-linking of utilizing this kind of carriers can prevent the desorption and the carrier can be entrapped in the alginate. So if you use alginate, it can serve like that. Okay, now if we utilize this carrier for the cross-linking reaction, so we can use glutaraldehyde for the cross-linking here. Okay, so this is the type of examples. So now let us look at the organic carriers from the natural sources. The type of organic carriers for the natural sources are, say here we can see most of them are polysaccharides as we have already discussed about. So let me take a color here again, change a color, and derivatives of the polysaccharides can be used. And this case of polysaccharide, they are pretty complex polysaccharides like the cellulose. So we can utilize DEAE cellulose and also CM cellulose, which is carboxymethyl here. Okay, so we can use both of this type of cellulose. We can use dextran as this matrix. Okay, so dextran is widely used for enzyme immobilization as well as protein chromatography, and also you can use other cellulose, other polysaccharides like agarose, starch and pectin, and also chitosan and all these things. We can utilize this type of polysaccharides in case of the production of organic carriers for the enzyme immobilization. But so we can see most of the part is consisting of this polysaccharides, but we can also use proteins like gelatin and so on. but this is very rare. Most of the part is consisting of polysaccharides. Now let us look at the organic synthetic carriers which are synthesized outside the system. It is not derived naturally, and the advantage of these synthetic carriers that they are highly durable. They are having mechanical stability as well as pressure stability. So what we do in case of inorganic carriers, we get pressure stability, but the bonding is not that much effective. In case of organic carriers, the bonding is very effective, but this is not that much stable. So we combine both of the ideas and produce the organic carriers, which the bonding is very stable and interaction is very positive. Along with that, it will also uh, be very stable mechanically, very stable, and also very pressure tolerant. Okay, so it is a very good capacity, and simple manipulation can produce this kind of synthetic carriers. And the examples are polystyrene carriers, polyacrylate, amber light carriers, like that. Okay, so we can utilize this type of carriers. And also resins are there, ion exchange resins. As we know, we can utilize this ion exchange resin for different types of purification processes, water purification and also different chemical purification purpose. We utilize this resins. Okay, polystyrene and polyvinyl acetate or acrylic polymers can be used as the synthetic organic carrier. Now the last point, the main advantage of immobilized enzymes. Three advantages are there. One is that easy separation from the reaction mixture, because if we take enzymes uh, just in the reaction mixture, we need to purify it, we need to extract the enzyme out of it. It is very difficult. But if we immobilize it into a solid matrix, it is very easier to take out liquid from the solid and extract solid from the liquid, right? So this is a very easy for the separation. And also it blocks the enzyme loss, so very minimization of the enzyme loss can be achieved utilizing the immobilized enzymes. Second important thing is that we can reuse the enzymes. So as we can reuse the enzymes again and again, having a minimum loss of the enzyme, it will lower the total production cost. Okay, so this is very important. So it is economically very important. Third point is that ability of the enzymes to replace multi-standard chemical step. So if there are multi-standard chemical steps to be proceeded, we need to add the enzymes various times during the reaction. It will be time consuming, as well as the enzyme loss will occur. But if you utilize this immobilization technique, it can replace the multiple standard chemical steps by just one time addition of the enzyme. Then whatever steps will be done, enzymes will be there to perform those steps. Okay. Now the applications of immobilized biocatalysts. Now these were the the advantages of immobilized enzymes. Now let us talk about the application of them. Now the application are in these different fields, like the isomerization of glucose to fructose in high fructose syrup. We can use them to produce it. We can use them in the lactose hydrolysis in dairy products, because we know in dairy products lactose hydrolysis can be utilized in to produce various kind of products which serves us in many ways. So we can utilize this enzyme biocatalyst or entrapped biocatalyst in lactose hydrolysis reactions in dairy products. In penicillin acyclase we can also use it, and also in various kind of amino acid synthesis we use this type of enzymes or immobilized enzymes. So these are the importance of these enzymes, and that is the three advantage of utilizing the immobilization in enzyme. And that's it, and I hope this will help you. Thank you.